I know 5401 is delivered intramuscularly, so it's a DNA vaccine that is injected into a muscle, and also uniquely it's given with an electroporation. So it's essentially, in simple terms, it's an electric shock into the muscle, and what that electroporation does is briefly open membranes in cells, and then those cells, the DNA is driven into the cells, and the cells take up those pieces of DNA. But then it's been shown that then the cells efficiently produce the transcripts that those DNA plasmids encode with very specific promoters on those DNA plasmids. So it's a novel way of introducing antigen really into the tumor. And it is truly a vaccine, and the vaccine strategy in general is a way to try to get these educated T cells into the tumor, again, because glioblastomas in general have very little T cells within the tumor mass, and that's likely one of the reasons why immune checkpoint inhibitor alone isn't functional. One other interesting thing about this drug and then this trial that's going to be conducted in glioblastoma is that they are also using a DNA vaccine that's encoding IL-12. So IL-12 is a pro-inflammatory cytokine and really turns on the immune system in multiple ways using various mechanisms. One of the principal ways is it can turn naive T cells into what we call a Th1 phenotype of T cells, and also can turn on and sustain memory T cells. So using that IL-12 vaccine also as an adjuvant to the very specific tumor antigens that they're also injecting DNA plasmids for is a strategy to hopefully increase the antigenicity of the vaccine against the tumor. And in this trial, they'll be injecting both DNA vaccines intramuscularly and electroporating them. And also, the patients will be getting cemiplimab, which is a PD-1 inhibitor, so it's an antibody against PD-1. It works similarly to other anti-PD-1 inhibitors that are available either on the market or in development. Again, it inhibits that PD-1, PD-L1 signaling access in T cells. So of course, the PD-1, PD-L1 signaling access can attenuate the function of T cells. So this trial will combine their novel DNA vaccines with tumor-associated antigens as well as IL-12 to drive T cells into the tumor, and then in conjunction use a PD-1 inhibitor to then, when those those T cells arrive at the tumor mass, can be then turned on or at least de-inhibited so that it can go ahead and perform its function and hopefully be able to eradicate tumor cells. Now, the trial will be conducted in newly diagnosed glioblastoma patients, again, to try to treat these newly diagnosed patients rather than at recurrence, because we think, again, immune therapies need some time to take effect, whereas in the recurrence setting, patients really may not have enough time for immune therapies to have effect. And as part of this trial, they're giving standard therapy, specifically radiation and temozolomide chemotherapy, but the other unique thing that they're doing in this trial is stratifying patients based on their MGMT status. So MGMT is a potent prognostic marker in glioblastoma. So patients with MGMT methylation, they certainly live much longer than patients without MGMT methylation within the tumor. The other thing that we've learned about MGMT is it's a very good predictor of response to temozolomide chemotherapy. So patients with methylated MGMT will respond to chemotherapy, and those patients will live longer. Patients without MGMT methylation are much less likely to respond to temozolomide chemotherapy. So in this trial, they've stratified patients by MGMT status. Notably, the patients without MGMT promoter methylation will not get temozolomide after radiation. And one of the reasons they do that is not just because their MGMT status predicts they may not respond to temozolomide, but what we do know is that temozolomide can be very lymphodepleting. So it can be very immunosuppressive when you give patients temozolomide over an extended period of time. So in principle, what they're trying to do is limit the amount of immunosuppression that these patients get exposed to during standard therapy because they may counteract what they're trying to do with increasing the number of T cells that infiltrate into the tumor. So in the unmethylated MGMT arm, they're skipping the temozolomide. The other novel thing that they're doing in the study is shortening the course of radiation. There's also some evidence, and actually old evidence from the 80s and 90s, that prolonged fractionation, so many days or weeks of radiation, fractionated radiation, can be more immunosuppressive and more lymphosuppressive than shorter courses of immunotherapy. So they're really shortening the amount of time that patients get radiation from 6 weeks to 3 weeks, again, try to limit the amount of immunosuppression that patients experience during standard therapy while they're trying to activate T cells against the tumor. In the MGMT methylated arm, where patients can certainly do well for a very long time with just even standard therapy, patients will get chemotherapy, the full course of temozolomide chemotherapy that they typically would get under standard therapy. But on the other hand, what this trial will do is also shorten the course of radiation that they get as part of that standard chemotherapy regimen, chemoradiation regimen, from 6 weeks to 3 weeks, again, in an attempt to try to minimize the immunosuppression that people get exposed to in this trial. The major objectives of this trial will really be about safety and tolerability. These agents have not been tested in patients with glioblastoma. We really don't know what the safety profile is going to be until patients are actually treated, how they tolerate it, how they tolerate the injections as well as the electroporation. Secondary objectives will be to look at efficacy, so how long patients will live before their tumors progress, or the overall survival as well, but also looking at a number of immune markers both in the serum and hopefully within the tumor in post-treatment tumor samples to see if the immune system does get activated in a specific way, looking to see whether T cells get activated in the very specific antigens that they are vaccinating against in this trial. One of the eligibility criteria for this trial will be patients who have had a significant resection of their tumor. That won't be 100% of patients. Many patients are only able to get a biopsy, we call a stereotactic biopsy, where really just a needle can go in and get a sample of the tumor just to establish a diagnosis and get some genetic markers. For those patients, this trial wouldn't be recommended, nor would they be eligible. But really, I think for most patients with glioblastoma, they're able to have good surgeries where the majority of the tumor can get resected. I think this is an excellent trial. I think that it would be inclusive of whether or not patients have MGMT, methylated or unmethylated status. They will be getting what we consider standard therapy for newly diagnosed glioblastoma, so they won't be withholding standard therapy. I think if patients have an opportunity to be on this trial, I think it'd be a very good option.